So I'm going to switch back to the document camera and remind you that we outlined the cell membrane, the nuclear envelope, and the rough ER, Golgi, and the vesicles last time, talking a bit about the different proteins that are synthesized here and there. You'll find that information in greater detail in the slide set that I just referred you to. What I want to do today is to talk about first the cell membrane, then second, and of course obviously the proteins that are included in it. Then second, we'll talk about protein synthesis. And then third, we will cover the gene expression regulation. Expression and regulation, if you will. So let's start first with a drawing of the cell membrane at a high magnification. So basically, remember we drew the cell with the nucleus, nuclear pores, and I always think of this as a basketball with a wiffle ball inside of it. We all know the wiffle ball is, although the wiffle ball wall has to have 2 layers, I guess, but it's got all the holes in it. So that's how I envision it. And then we did the rough ER, free ribosomes, Golgi, and the vesicles. And we talked about housekeeping protein synthesis on free ribosomes and 3 types of proteins being synthesized at the rough ER. That is secretory proteins, obviously very important in endocrinology and neuroendocrinology. Membrane proteins, which obviously we're going to talk about in our discussion of membranes, and the proteins that end up in the lysosomes. So there's a couple different types of vesicles, and there's very nice illustrations in the slide set that I just referred you to. You can print those out, black and white, color, however you want to do it. To follow along with those lectures and to have for reference material if this seems a bit remote and you'd like to get a better picture of a cell in your mind than what I have sketched. So we know that the phospholipids are the type of lipids found within the eukaryotic membranes. The phospholipids have a polar head and nonpolar tails. I think I call them legs in the streaming video, but it's the same thing. In eukaryotes, it's not completely saturated in one of the fatty acid chains that's linked to this polar head. Therefore, it's shown as having a little bend in it. It's different than non-eukaryotic cells that have completely saturated fatty acid tails, and they're perfectly straight. Not that it makes a big difference for what we will discuss. So this is the lipid bilayer. So the polar head has a plus and a negative charge, making it polar. Therefore, it is hydrophilic. Oops. Is it PHY? PHI, isn't it? Yeah. Sorry about the misspelling there. Hydrophilic, it's ILIC. And the nonpolar tails, of course, create a hydrophobic biophysical environment to be safe at this time. So this is the lipid bilayer, 2 phospholipids, feet to feet, and the heads opposing each other. Now let's put this together into a membrane, because we know a membrane is not just 2 of these. And now I'm just going to draw the straight tails for the sake of simplicity. I'm going to leave a gap so we can put a protein in here. few more on this other side over here so that we can label this. So it's the lipid bilayer, and we are going to think of the intracellular versus extracellular compartments. And this creates the separation. So we said we had the polar heads creates a hydrophilic or partially hydrophilic environment at the borders with these aqueous, A-Q-U-E-O-U-S, right? These aqueous environments of the extracellular and intracellular fluid. So what this means is that the aqueous water-soluble molecules can push their way in here. They're not fully repelled from the surface of the membrane, and occasionally a little bit of water can slip through. It's not a lot, but just a bit can. So it's not a perfect membrane, but it is close to perfect as far as the lipid bilayer being exclusive of water-soluble molecules. And that's because of this core of the membrane, if you allow me to call it that, that is strongly hydrophobic or lipophilic, obviously. It's hydrophobic, so if an aqueous solute or solvent, the water, tries to push in a little bit, it's going to push it back out. But if a lipid molecule comes along, straight through. So it's very important in endocrinology, neuroendocrinology, where we're talking about steroid hormones that are lipophilic and water-soluble hormones, peptides and the sort, that are hydrophilic and in the aqueous solution. Because the lipophilic steroid types of hormones will simply pass through the membrane. So there won't be a membrane receptor for it because there isn't a need for one. The receptors will be inside the cell. For the water-soluble hormones, there will be a receptor, and the receptors can be made up in a variety of ways. And I'm drawing the proteins, all the examples of the definitions of integral protein that spans the full membrane, and peripheral proteins that are only exposed on one surface, either the inner or the outer. That's all covered in those supplemental lectures, and I certainly am expecting everyone to have some recall of that. So there can be these complex protein associations with the membrane. They can move around. There's a fluid mosaic model. There's a wide variety of things that can happen for these proteins. They can have binding sites, associations with other proteins that upon binding you get conformational changes so that you get activation of something and then something else happens in the cell. So that's a signaling mechanism that we're going to focus on tremendously in this course for the water-soluble molecules. So what we just described, where something binds on the outside of the cell, causes a conformational change through this protein and then maybe some other associated protein with it, a peripheral protein on the inside of the cell, then can do something else and triggers a mechanism for response within the target cell, the cell that's targeted by this chemical messenger out here. That concept is transmembrane signal transduction. So this extracellular chemical, it's also intercellular, between cells, but it is outside, so it's extracellular. It's water soluble. If it's lipid soluble, it's just going to slip right through. So specifically for the water soluble molecules, hormones, let's just jump right to it, right? But it can also be for neurotransmitters. Neurotransmitters obviously play a big role in neuroendocrinology. and we'll cover that in some detail. So this is the signal. This is the target. The signal does not cross through the cell, so it must somehow get its information transduced across the membrane so that there is a functional response in the target cell. So it is transmembrane signal transduction. And it becomes quite complex, and I like to introduce it at this level just to get everybody geared up again to think about it in that way. So before moving on, I just want to illustrate a couple of other membrane-bound organelles that we talked about. The rough ER would be the same with proteins, except with the rough ER, what we would have is cytoplasmic. And I challenge you to think about what is the difference between cytoplasmic and cytosolic. Cytoplasmic is all-inclusive. Cytosolic is only the true aqueous part, where the other suspended molecules and organelles are excluded. So if you were able to separate cytoplasm from nucleus, spin it down so that all the organelles went down, whatever fluid is left, that aqueous solution, that's the cytosol. So it'd be membrane-free and all that stuff. So it'd be the cytoplasmic, cytosolic side of the rough ER or smooth ER, and then the intracisternal. Face of that. So as proteins are synthesized, there are pore proteins that come together. Again, that's illustrated in the, much better than I can draw it, where the ribosome is going to be out here with the messenger RNA. And if this is a protein that is supposed to be secreted, or a membrane protein, or a lysosomal protein, when the first amino acids are added on with the peptide bonds, it's going to push through these proteins in the membrane of the ER, and ultimately the protein will be pushed into the ER cistern. And very nice illustrations in the slide set that I have posted. So if this is a membrane protein, what's going to happen is it gets inserted in the membrane and will stay in the membrane of the ER. That portion of that membrane has to be shipped ultimately. Remember that turn this over real quick. In the cell, we had the Golgi apparatus with vesicles coming up, forming the endocytotic vesicle, and then the rough ER somewhere over here, and the ER Golgi interface where transport vesicles bud off the ER and go to fuse with the Golgi apparatus. So if we just created this membrane protein here, it has to end up in a vesicle. The vesicle has to get into the Golgi. This is the transport vesicle here, and then the secretory vesicle has to have it within it from the Golgi, and then also within it when it fuses, and then ultimately it puts that membrane protein into the membrane, if that's where it's supposed to be. You know, the targeting of membrane proteins is a very sophisticated, complex system, and we're just focusing on putting a membrane receptor into the surface of this cell that can be targeted by some extracellular chemical messenger. So what I want you to think of is at the ER membrane, when we get messenger RNA there that is for producing a membrane protein, the membrane will just stay, the protein will stay right in the membrane. That chunk of membrane will end up getting shuttled all the way to the outside of the cell, or the surface of the cell, if that's where that membrane protein is supposed to go. We're envisioning it as one of the receptors that we just illustrated before. So with a secretory vesicle with that membrane protein in it, this is the inside of the vesicle, right? This is the outside of the vesicle. So inside becomes outside when it goes to the cell membrane, right? Picture this going to the cell membrane, fusing, and then eventually opening to the cell membrane. This goes away, and then ultimately this gets inserted. Can I redraw it? Right, so put a little asterisk on this. It would be that part there. So what was inside the vesicle becomes outside the cell. Just a little mirror image trickery related to that. Not that it has any real relevance for what we're doing, but some of the research articles that we look at might isolate vesicles and try to study the protein, and if they're looking for a binding site on the protein, they might not be able to get to it because it's going to be inside there in the vesicle, because it will have to be inside the vesicle to end up outside the cell to be functional. So if they didn't lyse the vesicles, they're not going to be able to get to that. So that's one of the reasons for introducing it at this point, because we want to be able to criticize research methodology. If blatant errors show up like that and they get negative results, we can interpret and explain the negative results. So we did the rough ER and the vesicle membrane, and that was what I wanted, the two examples that I wanted to discuss. There's another very interesting thing I can't resist talking about. If we think of the nuclear pore and the nuclear envelope, remember each of these black lines I'm drawing now are lipid bilayers. In many cases, this outer, I call it a leaflet. It may not be accurate, but I'd have to go back and look at all the literature to see what they call it. But this outer nuclear membrane is continuous with ER, actually, in many cases. And there are many proteins inside the nuclear envelope that bind to proteins related to the DNA to hold the heterochromatin close to the nuclear envelope so the euchromatin can be all spread out in the rest of the nucleus. Anyways, a lot of details about it, but it's fun to look at that kind of literature sometimes. So how do these membrane proteins, these anchoring kind of proteins, get synthesized and get into the membrane inside the nucleus and the nuclear envelope? Well, they're inserted into the membrane here, and then this membrane actually just migrates right along all through here, and then goes where the nuclear pores are, where it bends into the inside, comes around, and gets pushed there. So the membrane movement of not only the phospholipids but of the proteins is really tremendous within a cell, within the endomembranous system. The nuclear envelope is part of the endomembranous system. It's continuous in many spots with the ER. The ER is separated maybe by vesicles, the transport vesicles, but it truly is continuous with Golgi with the extracellular, the surface cellular cytoplasmic membrane. So it's just kind of an interesting thing. And these nuclear pore proteins are, of course, synthesized on free ribosomes out here, and then are delivered with permission to go there. These nuclear pore proteins have recognition sites on them. There's other companion proteins called guard proteins. So they stand there and they're on guard. If a protein happens to come by, bumps into it, if it doesn't have the proper sequence, a nuclear sequence, giving it permission or access to the nucleus, it will not be allowed in. It's not pushed away, but it just will, whatever brought it here will bring it away, I guess. Brownian motion, or I don't know, jumping up and down. Does that move our cytoplasm? Who knows what makes stuff flow around in the cytoplasm if it isn't caused by the cytoskeleton. But a protein that is permitted to come in, like say a ribosomal protein that is based upon the assumption that ribosomes are assembled in the nucleus, comes up, it has permission, and therefore it can come through. There is definitely a protein from the cytoplasm that can enter the nucleus and will bind to portions of the DNA relevant to another part of our discussion, and those are transcription factors. So these are proteins that, once they are activated, they go through a conformational change, and we'll see examples of these throughout the semester. They undergo a conformational change. They now have exposed the part of their protein, their amino acid sequence that gives them permission to come in. Guard proteins say, okay, you have privilege to go through the pore, and will bind to the DNA near genes, called a spot called the promoter region that we're going to illustrate in a while, and can affect the transcription of DNA. So any protein that can bind to DNA and affects gene transcription is called a transcription factor. We'll look at many of those throughout the semester. Changes in gene expression are extremely important in endocrinology and neuroendocrinology. Prime example is a stimulus to an endocrine cell causing the secretion of a peptide hormone also has to bring the stimulus to the nucleus to synthesize new hormone to replace what you just released. So it's a homeostatic mechanism. The stimulus to an endocrine cell must trigger both synthesis and secretion. Secrete because that's the signal to the endocrine cell and the response of the endocrine cell to that stimulus, and synthesis because you have to replace what you just released. So the next thing, we wanted to talk about the protein synthesis. So we've seen that repeatedly, and we'll just get to it again. In a short while, we'll talk about the generation of messenger RNA, but we know that only mature messenger RNA, obviously this is the nucleus and this is the cytoplasm, and this is our nuclear pore again. Only mature messenger RNA has permission with the internal guard proteins to exit or translocate out of the nucleus. Translocation is the term for this movement from cytoplasmic to nuclear or nuclear to cytoplasmic. So a cytoplasmic molecule is translocated into the nucleus. A nuclear molecule is translocated into or translocates into the cytoplasm. We know that this messenger RNA has a sole destination as far as the organelle that it's going to interact with, and that's the ribosome. I'll roughly illustrate it there. There are the two types of ribosomes, as we mentioned, free, or I'm going to use the archaic term, and forgive me for that, fixed, or very close to the endoplasmic reticulum. So fixed is just an easier way of saying the ribosomes that are going to interact with the ER. Actually, there can be multiple ribosomes that jump on this message, and each of them begin to produce a peptide. So that's well established. It's probably a bit limited just with physical space if it's going to be associated with ER. Here's the protein pore, the ribosome that we had drawn before with the protein being produced entering the ER lumen or cistern. So those are the two options. Again, free ribosomes, any type of general housekeeping protein, and we'll encounter many. Many that are in that category of general housekeeping actually are very important for intracellular signaling. So extracellular signaling is through transmembrane signal transduction is converted into intracellular signaling, and we'll see there's multiple pathways. You activate this, activate that, then you've got a protein that's activated that now can go into the nucleus, and so on and so forth. So that's not the typical type of housekeeping that we think about, but it's still in that category. When I think of general housekeeping, I think of mostly the carbohydrate synthesis, types of proteins for that level of metabolism. But the proteins and enzymes responsible for membrane synthesis are included in that as well. So I don't get into the details of all this. I can't illustrate it. There's a little bit more information in the slides and streaming video that I mentioned. It's not our intention to get into a great deal of detail here. The messenger RNA is different, and the first part of the peptide is different, and we'll discuss that in later lectures. It's the first part of the messenger RNA that is translated into the protein, the first part of the protein that actually causes the interaction with these proteins in the ER to make these ribosomes fixed now and to target this protein into the ER. So there's a signal sequence and a variety, a couple of different terms that apply to that. So it's actually the messenger RNA that determines where the ribosome will go once binding to it, whether the protein will be produced and sent into the ER or just produced in the cytosol and be released into the cytosol when it's fully produced. So once a protein is produced, there are many things that can happen to it. So you've got this long chain, and there's the primary structure, secondary structure, tertiary structure, quaternary structure of proteins. I'm not going to get into that. We'll look at examples. Most importantly for us, there can be a variety of post-translational modifications of proteins. One is folding. There is some of that goes on with hormones, and we'll see examples. Disulfide bonds are allowed to be formed within the ER lumen, so we can get a folding. Insulin is a very interesting one. It folds back, gets cleaved in a couple different ways. There's a couple of other interesting ones where that becomes very important. For biotechnological methods of producing proteins like human insulin and stuff like that from bacteria, it was very important to demonstrate that the appropriate folding and cleavage would occur so you could isolate bioactive insulin. So I'm not disregarding it entirely. But another one is glycosylation. So adding a carbohydrate chain to a protein is very important functionally. Many of the membrane receptors, many of the membrane proteins are glycosylated. Many of the membrane receptors are glycosylated, and it's near or at that glycosylated site that the hormone or ligand is going to bind to the external or the extracellular surface of that protein. And then the third important one is cleavage. So with cleavage, you can start with the initial translated protein, and it can get chopped. The terminology, we'll see it in more detail. There's a pre-pro hormone, gets cleaved, then it's a pro hormone, it gets cleaved, and then you've got the hormone, and if it underwent everything else it was supposed to, some hormones are also glycosylated. It finally now is, maybe there's a disulfide bond in here, and a little carbohydrate on it. It now is biologically active. Went from pre-pro hormone to biologically active hormone. It's now ready to be released into the extracellular fluid compartment to go and find its target, bind to the receptor, and cause the effect. Well, now let's get down to the gene. We've got about 10 minutes to talk about this. I may spend just a bit of time at the beginning of the next lecture to add a few more points, but I'll get the major ones expressed now. So and as well, I cover this in quite a bit of detail in the lectures. So we know there's the double-stranded DNA in the helix. Can I do it? No. And it has two strands, the sense, they're anti-parallel, we know that, and the antisense strand of DNA. It is the antisense strand of DNA that is transcribed. So the messenger RNA is actually the sense. We know it's a different type of nucleic acid. I'm not trying to overlook your background knowledge. But the sequence for the messenger RNA is actually the sense sequence. So there are methods, once you end up with the messenger RNA, there are methods of using antisense cDNA to bind, to hybridize to messenger RNA, either to identify it or to block it, and a variety of things like that so that it can't be translated. So it's important to understand that the antisense strand of the DNA is transcribed to make the RNA, and the RNA in that sense then is equivalent to the sense strand of the DNA. So to get this DNA to bind to the messenger RNA, you would have to generate the antisense sequence. I learned that the hard way one time when I called a company to order some cDNA, and he asked me, and I was like, man, it's a long time since I thought about that. Which strand of DNA gets transcribed? So what do other people order? He's just laughing at me inside. So I started reviewing again way back then as a postdoctoral trainee to catch up on all of this. So we know that these two strands have to be separated, so there's a loop has to be uncoiled, and then a loop that allows for proteins to come in here. If this is the antisense strand, then proteins are going to be in here to open this up. There has to be a spot where the RNA polymerase can bind and move along to produce the RNA. So let's just jump away from that. There are better illustrations of that, as I said, available on the slides that I've provided, and certainly in other textbooks and illustrations. Let's just illustrate a gene as we would in an introductory genetics course, where this is the 0 and this is upstream. Sorry. Downstream, and this is upstream. So there's and this is the start, or the 0 location. I'll just use this term start site. Those of you that are have already taken more sophisticated courses know that that's a generalized usage of that term. So from the start site, there's upstream sequence and downstream sequence, and we're going to spend a lot of time talking more about upstream sequence because this is the promoter region for a gene. And everything we talk about is for regulation of gene expression really has to do with promoter region. The downstream portion has two components. Anybody remember what those are? There's this portion in here and then this portion here. Exon and the intron. So the intron are intervening sequences. They are transcribed but not translated. So from this, the RNA polymerase is going to bind here. I'm not going to get into data boxes and all that kind of stuff. We don't need to worry about that. Plenty of other courses where you get that. RNA polymerase binds here, moves along. As it moves along, it generates the initial transcript. This is not mature RNA, so now we have to talk about RNA processing. So there are at least 3 examples of RNA processing. There is the 5' cap, and that's what, methionine? Nobody's objecting, so if I'm wrong, you're learning the wrong thing. So double check on this one. I'm not sure my memory's working so well right now. But there is a 5' cap, and then there's the poly-A tail. Oops, poly-A tail. So if we draw our initial sequence here, poly-A gets added on to the 3' end enzymatically, and then there's a nucleic acid added here, which for the moment I'm thinking is this, but it may be wrong, so don't trust me. And then the third is splicing. So along here we have somewhere representing the exon and the intron, exons and introns from the parent DNA of the gene, right? So what happens is with splicing, you actually get the splicing out of the introns, and then so there's a cleavage, a clipping and a removal, and then there is the actual splicing back together. I'm trying to think, there's a term for hooking the nucleic acids together. I'm not recalling it right now. The exons stay. Now this isn't always true. For one gene, you can have what is called alternative splicing. And this becomes important in biology. It's known more and more that one gene can produce more than one protein isoform due to alternative splicing. So if there's 3 exons to start with, we say the exons stay. Well, maybe something changes that and only exons 1 and 2 stay and exon 3 gets cleaved out, or exon 1 and 3 stay and exon 2 gets cleaved out. It's messenger RNA from the same gene, same sequence. We know that sometimes you can have 2 genes for the same protein, but the DNA is slightly different, so you get isoform 1 and isoform 2 of the same protein. But in other cases, you can have 2 isoforms or more of the same protein from the exact same gene. That's because of alternative splicing. Now, in the last couple minutes, I just quickly want to talk about the promoter region. So here's our start site. Here's the promoter region. So we're at 0 here, and that's upstream. We know there's a TATA box here somewhere. And these are all numbered. So typically somewhere within the first 1,000, let's say, definitely within the first 1,000, there are DNA sequences. They're usually in pairs, and they're capable of binding proteins, and they're capable of affecting gene transcription. So what happens is when a protein binds here, you get a response. This is an element. So these are called REs, response elements. So for example, for the glucocorticoids, where the glucocorticoid, let me spell this first, I can't talk and spell glucocorticoid at the same time. There is a GRE. So if we make this a GRE, glucocorticoid response element, then 2 ligated, the glucocorticoid has to be on it, 2 glucocorticoid receptors that have been ligated, or they have the glucocorticoid molecule itself bound to it, they form a homodimer. We'll talk about homodimers, heterodimers, and all that stuff as we proceed. These are now changed conformationally so that they stick to each other. They're ligated, they can stick to each other, forming the homodimer, and their DNA binding domain is exposed. They now can bind to this specific sequence, and at some point you'll memorize that GRE sequence to answer a question on an exam. You may never remember it again in your life, but anyway, we do things episodically in life, and it feels good, I guess. And once bound, changes the conformation. Maybe it causes a greater uncoiling of the DNA, making it more likely that RNA transcriptase will bind here in more frequently. Therefore, the increased probability of a more frequent binding of RNA polymerase, so an increased probability of a more frequent binding of RNA polymerase is the definition of increased gene expression. Conformational change of the DNA upstream of the gene that's transcribed and translated after RNA processing can either increase the probability of the frequency of binding of RNA polymerase or decrease it. If you increase the probability of binding and frequency, you increase gene expression. If you make it more difficult, decrease the probability, then you decrease gene expression. So in cells where glucocorticoids are promoting of cell survival and growth, those proteins, genes, very likely on their promoter region have the GRE, and when it is ligated, when that GRE is ligated by the homodimeric receptor, ligated receptor, it's going to increase the frequency or the probability of the binding of the RNA polymerase there, increasing gene expression. If we're talking about the pituitary gland, where if this is the ACTH gene, adrenocorticotropic hormone that drives the synthesis and secretion of the glucocorticoid at the adrenal cortex. I don't want to jump too far, but just to get that, this must feed back on that, have a negative feedback. So if this was the ACTH gene, then negative feedback should decrease synthesis and decrease secretion. So we're going to have a conformation change on the ACTH gene such that the glucocorticoid response element effect is to decrease the probability of frequency of RNA polymerase binding to the ACTH gene and transcribing. So I know I turned it into a whole bunch of different words, but what we have to keep in mind is, and it's so easy in this type of a course where we're talking about molecules going from here to there. You know, we picture this molecule being released and under its own desire going out and looking and going, oh, there's my receptor, this is where I'm supposed to be, and this is what I'm supposed to do. And we know that that isn't true. If I release it here, I basically just drop it out into the extracellular fluid, and it somehow gets out there and happens to be near its receptor, and when it hits, then it can produce its effect. So it's diffusion, it's bulk flow, the convection flow of interstitial fluid, getting it into the plasma. So diffusion from higher concentration to lower concentration at the edge of the cell into the plasma. The plasma is always washing by, so it sustains that gradient of high near the cell to low in the plasma. Plasma helps to bulk flow to get it out so it can diffuse out. Now the gradient being different, higher in the blood, lower in the interstitial fluid, gets out there and is just in the solution. So I'm inside the cell, here's the receptor. Out there in the interstitial fluid, there's either a high concentration of these molecules randomly bumping into my membrane or a low concentration. The more molecules that are out there, the more likely one is going to successfully bump into the receptor just right to do the lock and key fit and activate a receptor. So it's the best time now is to remind ourselves that even though we say it starts here and it goes there and it binds to its receptor, it's much more complex than that. It's not self-motivated. Molecules don't have any way of propelling themselves around through the body. So I appreciate your time and attention, especially the extra few minutes here, and have a nice weekend. We'll see you on Monday.